Okay, good evening, everyone. I'm Sue. I'm the host tonight for Taxonomy Tuesday from NJMA. Tonight we have a short presentation on one of the sections of Amanita by Hunter. So Hunter, if you would like to start.
Yes, thank you. So Amanita section Vaginatae is one of the 9 sections of Amanita that we now have. And Quo on mushroomexpert.com kind of simplifies the general characteristics down to just 3. And the first is strong lines or striations. The second is no annulus, ring, or skirt. And the third is a sac-like vulva. And we'll talk about each of these for just a couple minutes. But what I want to say is that it's pretty easy to identify to the section. And so if you're not familiar with the section, I think you will be by the end, and you'll be able to at least do that. And that is all I can really promise here. But let's continue. So these strong striations that I just mentioned, you can see in these various species of Amanita section Vaginatae, and they take up at least 25% of the cap diameter. That's kind of the requirement to put them in the section. And you can see, although the colors are pretty drab, there is a wide range of colors ranging from steely gray to brown to olive green to what they call Chinese yellow, curry, orange, brown, and true brown. The nature of the striations are tuberculate striate, which is really interesting. So they're created by ridges that you can see pretty clearly in this specimen. So ridge lines create the striation.
Excuse me, Hunter, I just want to interject something here.
Yes.
Your photo does show tuberculate striate cap margin, but the word tuberculate refers to the little bumps or pimple-like things that are on or in between the striations.
Yes.
And I don't think that all of, or even most of the section Vaginatae species are tuberculate or maybe at least strongly tuberculate.
Okay. Thank you for
Another
that.
thing I'll say is Michael Quo, I think, is wrong when he says that a sacate membranous vulva
Yes,
is
I'm
a
going to get
key
to that.
character. Yeah, you're going to get to that. Good.
Yes, I'm going to get to that. Yeah, and but on this, I'm glad you corrected me, because I wasn't positive. It's certainly more obvious in like this specimen and some of them, and so thank you. I think that you're correct that they're not all tuberculate striate, but some species are, and I'm sorry about that. And thank you for that. And so some other cap features that we can look for are different zones of color, like you see on the left, where it's cream on the outer edge of the margin, and then more brown to light gray to dark gray. And that's indicative of a couple different species. One is Amanita fuscozonata, another is Amanita luzernensis. And then another cap feature you might see is this dark center, like you see in the specimen on the right, and that might be a species called Amanita magna, or one they called Amanita cyclops. But I don't think that that's I think that's been deprecated. In some species, there'll be just one patch of velar material left on the cap, and so you might see some in the section looking like this. And then there is Amanita rhecopus and Amanita texas aurora that have these gray veil remnants on the cap. And so this is actually pretty easy to identify. It's at least one of the two, but it would take DNA testing to know which one of the two. So generally, these members of this section are 3 to 6 inches tall. But on the West Coast, we have the king of grisettes, Amanita pachycolia, which can be up to 10 inches tall. So lacking a ring or annulus was the second general characteristic. And so you can see in these members of section Vaginatae, there is nothing between the cap and the vulva or the base. No ring, no annulus, no skirt. And that's called X-annulate. And so, yes, Quo says they have a sac-like vulva, but really the vulva on these this section, depending on the species, really ranges from strangulated on the far left, friable, which means like breakable, brittle, falling apart, which I think the second specimen may represent. It looks like it's not going to hold up, but to the point that when you excavate one from the ground, the remnants may be left in the dirt. You may not be able to gather them all. To less constricted, and then to far less constricted on the right. And some species, as they get older, the vulva material will turn gray or even black, or sometimes it'll have brown on the edge, brown and red kind of staining on the edge. So their gills are white, like the majority of Amanitas, and tightly packed, and short gills are abundant. For the most part, they're free gills, but two adenate, two attached at times. And so originally there were just two. So Amanita was divided into two subgenuses: Amanita subgenus Amanita and Amanita subgenus Lepidella. And when you applied Melchers to members of Amanita subgenus Lepidella, like this Amanita citrina at Michael Quo's website, and as you look at the spores, you can see that they're amyloid, that they reacted and turned blue. And
We don't see that, Hunter.
oh,
We don't see the citrina.
Okay. Oh, that's weird that didn't work out. That might be problematic. At any rate, some of my transitions may fail us. We will see.
Sometimes they're slow on Zoom to take.
Okay. Well, and you've kind of all seen that before, so I'm just going to describe it here. So members of subgenus Lepidella, the globose spores would react when you put Melchers on it and would stain blue, whereas those in subgenus Amanita had no reaction. And so Amanita section Vaginatae is one of the three sections originally in subgenus Amanita. And it's my understanding now that Amanitas have been divided into three subgenuses. And so this is kind of old school we're talking, but at any rate, your Amanita section Vaginatae should be in amyloid. The spores should not react, should not turn blue with Melchers.
Hunter, the new taxonomy does not affect those three sections very much.
Good,
I
okay.
think there might be some like what do they call them? Sectoid species that are in subgenus Amanita. They may have created a section for those. I forget, but most of those are in Australia, I believe. So they're really of very little interest to us.
Okay, well, that's good. This is one you might possibly misidentify as a member of the section Vaginatae. It's got the kind of drab colored cap. It has the striations. But once this matures and the cap opens, there should be a veil under it, and it's a member of the section Ceciliae, Amanita spreta, and the vulva too would give it away for those familiar with that section, the lobe shapes of the vulva. But otherwise, you could see how someone might mistaken it for Amanita section Vaginatae. And then this Amanita virginiana, Dave alerted me to, and I'm gonna see if we're gonna be able to go to his observation.
Yeah, there's actually two other species names and possibly a few cryptic species that are unnamed that look like Virginiana. Subvirginiana and Pachiosperma are
Oh, okay.
small Caesars. I've been calling most of the ones that I've found, and I found maybe like 4 or 5 times these little Caesars. Okay, yeah, mushroom. Anyway, they they're small. They look a lot like Vaginatae, and the rings, the partial veil is kind of fragile. It falls off often,
Yeah.
so then it becomes a real problem.
Did your observation become visible to everyone?
Don't
Are-
see it. No, we're still
We're
looking at
not.
strata.
Shoot. Okay. So yeah, my transitions are not working. Hold on one second.
It's probably an issue with how you shared the screen onto. You probably shared the PowerPoint, but not your whole screen. So we can only see the PowerPoint.
Okay.
You'd have to reshare.
Should I reshare? I could. Yeah, I probably better. So when I just share again, I just
Usually-
choose the whole screen on top there,
Yeah, usually the option for the whole screen is on the left.
entire screen. Thank you so much. Okay, can you see it now?
Yes.
Perfect. Okay, so this is Dave's observation of Amanita virginiana, and I mean, based on what I've shown you, we've got the drab colored cap, the strong striations. As we look at the stem, we don't see an annulus. So gee, I think it's the gills are free. I think it's it looks a lot like something in Amanita section Vaginatae. But actually this had a annulus, and it just had gotten washed off or fell off, and so the Amanita virginiana is not in the section.
Could you show the species proposals for that one? Because I thought at first it was section Vaginatae. I had a discussion with Ratulas, and sort of together we decided it was probably Caesarea, and I think it ended up getting sequenced or it was sent. It was sent to Rhett. Rhett is Ratulas,
Yep.
and he examined it. And there are other features that are microscopic that separate Vaginatae from Caesarea. I honestly don't know these features very well.
Yeah, the spores are here, and I mean they do look different.
That's a lousy spore picture. I was
Okay.
trying to take pictures through my new microscope at that time, which is a binocular scope, and the pictures just weren't very good.
Okay. So, so it's not that hard to ID to section, but still there are, you know, some things to be cautious of. And so in that case, when mushrooms have a annulus, we call them annulate, and and you can see the strong annuluses on the first two specimens, but the one on the far right is in Amanita section ceciliae, and you can see how kind of weak the annulus is, you know, in comparison to the destroying angel, and so that these can fall off, these can wash off, and it's important to understand that, because the Caesars also have striations as well, but they tend to be much more brightly colored. And so I wanted to just mention a couple notable species to me, and you know, sadly, notable means like I kind of get them, you know, it's like I sort of get these ones. And so I'm going to share them with you. And you know, what I realize is how regional this really is, this section in particular, maybe Amanitas in particular, but it seems to me like the specimens in the Northeast are really different from those in the Midwest, and you know, entirely different from those in the West, and then also different from those up in those listed on Micro-Quebec. So I'm just going to share a couple I'm kind of familiar with, and then this is just, I'm just referring to it as Amanita stirps fulva, meaning it looks like fulva. And that's another thing I noticed when going deeper into Amanita section Vaginatae. There's a lot of stirps, groups, series. It is really, so many attempts and efforts have been made to try to organize and classify, but there's so much more work to be done, apparently. So this is Amanita fulva. It has this, it's called the tawny grisette. It's known for this orangish brown colored cap. There's, you know, some variation to that. And I had a couple more examples, but I don't know what's going on with my little slideshow. But that's kind of a pretty typical looking Amanita fulva. And then, so the section, the common name is grisette, and it was, and the type species for section Vaginatae is a gray mushroom, and it got the name grisette because in the late 1600s and early 1700s, French working class women wore these, who worked in textiles, wore this gray fabric, this dark gray form of dress, and that's what it was named after. But it also had some sort of kind of negative connotation, sort of derogatory, and it kind of at certain times in history at least just meant like a lowly person. And I think that's kind of fitting for the group in terms of the culinary experience with mushrooms, although the majority of the section is edible. I think they're just kind of, you know, so they're just, you know, they're not, there's maybe just one or two really choice edibles in the group. I think, you know, people always like go with the definition of grisette, but I think probably it was people, and it's just speculation, but would probably be like, yeah, what did you find any mushrooms out there? And they would say something like, yeah, I found some grays, like we talk about morels and, greenies and grays and I like to imagine that's how it goes in France. But anyways, in terms of choice edibility in the section, this is the #1 choice edible, Amanita villosa. It's primarily found in Northern California, but Washington and a couple other places in the Pacific Northwest. It is highly prized choice edible. It has these white patches on the cap and this sort of salmon colored, beautiful cap, large stem, large vulvic sac. Sadly, confused with the death cap and Amanita ocreata, and people have been poisoned when they had wanted this mushroom. And then I wanted to just point out one more, and this is one that Dave had alerted me to, Amanita penetratrix. And it's loading. This one has a rooting base and a really pronounced umbo that I believe aids in breaking through the earth upon kind of emergence. But this is this is very, very common in Connecticut, like very common apparently. And so I kind of wanted to just end on that note, on a mushroom that people here might have come across, and I'm hoping Dave can share some more with people. And I think that's about all I got.
Thank you, Hunter.
Yeah, thank you.
Yeah, good stuff, Hunter. Thank you.
Thank
Yeah, good job, Hunter. Just a few comments.
you.
Amanita rhacopus and presumably also Texasorora, if I'm saying that correctly, typically do have gray warts on the cap, but the warts often wash off, and then they're a little bit tricky to identify. Second, I don't think that Amanita cyclops is deprecated. I think it's just still a non-prov. I don't believe that there's been a name proposed to replace cyclops. I'm not sure about that. Your photo that you called a strangulated vulva did show a vulva that was adhering to the very bottom of the stipe base, but the rest of that vulva was still rather membranous and sac-like. When we talk about the strangulated vulva, the strangulated amanitas, those are the ones like Rhacopus, for instance, where the universal veil basically disintegrates from the bottom up and leaves only a very thin deposit of grayish UV material on the upper part of the stipe base and nothing else. You don't see anything else. It's all gone. And so the reason why it's strangulated is because it's just this thin ring that's going around the upper part of the stipe base. For the species formerly housed in section Lepidella, I think you said that they have globospores. It was probably just a slip of the tongue. I don't think there are many species in former Lepidella that have globospores.
No, just
I
Citrina.
Citrina is in Validae. And quite a few of the species in Validae have globose to subglobospores, mostly subglobose.
I was talking about subgenus Lepidella.
Oh, I'm sorry. Oh, my bad. Yes.
Yeah,
If you
get
into
only
former
Citrina.
section Lepidella, which is now Roanokensis,
Right.
most of those I think have ellipsoid to elongate spores. I'm not certain of that. I'd have to review. Rod told me once that there's a species of Vaginatae found in Africa that's dangerously poisonous. So this should at least inform the potential experimenter that there might be some um, 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 reason to exercise caution.
Yes, and Amanita crocea too, they say like it's cautionary, they're cautionary about too, I believe.
Yeah. Well, you brought up Amanita sprata. Gary Linkoff hypothesized that the name sprata, which means hated, was placed upon this species because people had mistaken it for people had picked Amanita phyllaides thinking it was sprata and probably died. So
Oh, wow.
sprata and phyllaides look a little, they're superficially similar looking. I mean, there are differences, but I can see where that would be a mistake. The Amanita penetratrix is a provisional species, and it's really not anywhere near being settled. It's a group, really. So Rod was calling it, I think, series penetratrices. So there's like penetratrix, penetrans, pseudo penetratrix, I think is one of them. But they all do share the traits that you described, being the deeply rooted stipe and the hard, notable central umbo that helps push the fruit body up from deep within the ground. And lastly, I'm going to say that I learned something because this Western North American species, Amanita villosa, I had always thought that it was a Caesar. And I double checked. I looked. So as far as I can tell so far, you're right. It's a species in section Vaginatae. So thank you.
Thank you, Dave. Thanks for your help this week. That was good.
Yeah, thank you, honey. I was trying to find out if I had any observations of Vaginata other than the ordinary ones. Can you tell me where the species rooseveltensis is in the Vaginata section?
I'll take this answer.
Okay.
Yes, it's very common species in section Vaginatae that seems to occur quite a bit in New Jersey and Pennsylvania. And
Yeah,
we're
I
not
noticed
sure
you
of
had
the range.
hundreds of observations, and I have one
Yeah,
lonely
well,
one in New
I
Jersey.
probably have 100 on mushroom observer because it grows in my yard.
Okay.
Rod Toulas made me a co-author on this species. It's a non-prov. It's going to end up being replaced by something else unless somebody eventually decides to try to publish it. It's a pretty distinct species, and as you suggest, it looks like, morphologically, just looking at, looks like Vaginata which is European, and Rod says we don't have it here.
We don't have it here?
Rod says we don't have it in North America. So DNA seems to be saying we don't have it.
So these are all the ones that you have, lots of them.
Lots of them, most of them from my property, and a few from south of where I live, a few from very close to where I live. This species seems to favor lawns with oak or hickory and older trees. That has been my expectation. But the thing that separates this species from most of the other common species in section Vaginatae is the spore shape. Most species in section Vaginatae, with some exceptions, have globose to subglobose spores. They're really very round. Amanita rooseveltensis has broadly ellipsoid to ellipsoid spores. And so some of them are, when you scope the spores from a print, you're going to see some that are like standing upright, so they'll look like they're round, but that's going to be a low percentage. Many of these observations you're seeing here, most of them are mine, If you
Yeah,
if
I
you
know.
click on the observation and go through the photos, you'll find some spore photos.
Yeah, it'd be
yeah,
hard to find though. We should get so many
there
of
we
them.
are. There's one that's like, I guess, one of the headline photos. Go down a little bit. There's one that's like, that's
Okay,
spores
gotcha.
right there. Yeah, they are ellipsoid, broadly ellipsoid. They're not round, and that was the thing that really got me thinking, like that I should send this material I was finding in my yard. And by the way, for me personally, this species represents a sea change in my interest as a mushroom enthusiast. When I first moved into this property where my wife and I live here, in Northeast Pennsylvania, I was all psyched about finding like Boletus edulis and Lexinum and chanterelles. I didn't find any of those things. Instead, there were like many, many, many of these things that I could see from my front porch. And they kind of got on my nerves for a while, because they're just like these silly little amanitas. Well,
Is this one
I got
edible? This Roosevelt one?
I ate one this summer, and it was pretty good. I mean, not a delicacy, but it was pretty good. Fulva is actually pretty good. Fulva is a pretty good edible, in my estimation. But anyway, yeah, I've eaten these once, no problem. They tasted okay. I didn't get sick or anything. But that's the thing that really sets this species apart, at least in this region, right? If you go into the Midwest, there might be some other gray a species from section Vaginatae that has ellipsoid to broadly ellipsoid spores, and it might be judged as being distinct from this one. There are a few of them, I believe. I think one of them is oh geez, somewhere in Louisiana. It was also named after a location. Roosevelt, New Jersey is the namesake of this mushroom. This grows in Rod Tillis's yard. He's got oak trees there. So this was one that really caught his attention early on because it grows in his yard. So he named it after where he lives.
Okay, thank you, Dave. Is there anybody else who would like to share species of Vaginata section that hasn't been shown yet?
I have a question.
Okay.
I was wondering about pakimpondensis. I saw that Jason might be able to say something about it too. For the New Jersey Forest Service, that they just found it on their land, and now it's been found in several places in the New Jersey Pine Barrens. I'm wondering first, I've been asking around if there are any fungi that are endemic to the New Jersey Pine Barrens, and I'm thinking this might be one. So far, I've heard people say they don't think there are specifically, but this one might be a candidate. And then I was also wondering, can you tell it apart macroscopically from other in that section?
I'll let others answer the micro part of that, but I just had one sequenced, and it matched for pakimpondensis. The match was from Florida.
Oh,
So yeah, so I don't think it's endemic to Jersey,
okay.
at
Okay.
least.
Yeah, I forget where I saw there was one other state. I think it was probably Florida. I forget where I saw that record. But
Okay,
yeah,
interesting.
that New Jersey Forest Service Facebook post was highlighting Brandon's discovery of that species on one of our trust preserves, but they didn't do a very good job at specifying that it wasn't the Forest
I
Service
see.
that made
I
the
see.
discovery.
Interesting though, still interesting. And so how can you tell it apart
Wait,
macroscopically,
what?
or can you not?
I think only because I've asked this here before. I think it requires sequencing, but Igor knows more about it than I.
Okay. If that's the case, then that's good to know, so I don't waste a lot of time.
Does this one have globospores or ellipsoid spores? Does anybody know?
Well, we could check Rod's site if it wasn't down today.
Yeah, that's what actually what I was trying to do that today. I noticed the site was down too.
Yeah, it was up yesterday for like a half an hour.
Okay,
and then
I don't
it
want to waste any more
disappeared.
time on that. I can check that out myself later. Thank you.
That's okay,
Yeah,
but you
thank
know what? There's a little bit more to your question, though, I believe. There are a number of species that are found in the New Jersey pine barrens that seem to have be also found in various places along the eastern seaboard, but
Right.
mostly, coastal. Some of the same things you'll find in Cape Cod.
Yeah, I was saying
Um,
I went up to when I went to I did go to that Anama, nymph for the first
Nymph, yeah.
time. And yeah, it was amazing to see so many of the same species there. It really got me thinking about pine barrens fungi, period, not just fungi that grow in, conifer forests, but specifically like outer coastal plain. I was wondering if there are any endemic to the pine barrens, period. And then I was also thinking specifically about New Jersey.
I think there are probably some species that are rarely, if at all, found outside of the pine barrens type habitat, which is a sandy soil and specific types of pines and
Right.
oaks mixed in.
Right.
Yeah, and I'll throw in. I think I mentioned it to you, Kiara. I have family in the UP of Michigan, and in some of those Upper Peninsula, Great Lakes pine, Great Lakes adjacent pine forests, we find very similar things that elsewhere in the world would be rare, and it's a very very similar setting.
Interesting.
And one of the books that Nina always likes to recommend is The Mushrooms of Cape Cod by
Yeah, I love
Grisette.
that. I use
Yeah.
that for the pine barrens. I think there should be a New Jersey version of that. Come on, everybody. Got to work on that.
It has to be somebody younger like you.
I don't have the wherewithal, but I would love to work on it with anybody.
Yeah, I'll send some photos.
Let's all work on it together. We'll do a side project sometime.
Yeah, but stick with
But
photos. As soon as you attach the name, it's going to be out of date.
I know.
I know Igor's here, and I want to hear his thoughts on that pakimpondensis discussion because, I was going to share an observation later on, and maybe we can talk about it then.
Yeah, I can show my own observation that was sequenced, and I have some good photographs to show the morphology of pakimpondensis, although it's only one specimen, only one mushroom. I can probably put it in the chat and somebody can share it on the screen.
Yeah, cool.
So,
And I don't know if you heard, I just had a sequence come back matching pakimpondensis too.
yeah, I heard that. So
That's so cool.
It's in the chat, so if somebody can open that and
I'll
share.
open it. I'll open it.
Is that the farthest south we've seen it on the East Coast? Have we seen it anywhere else?
I don't know. It's not a commonly found species. It could be coastal plain species.
Okay. Oh, Florida, was that outer, I don't know if all of Florida is outer coastal plain, is it? You found in the outer coastal
Florida
plain?
is coastal plain mostly, unless you're talking to the upper part of the panhandle. So it has a gray cap and plenty of graying vulva. I'm not sure if it's gray from the get-go or it becomes gray and gets darker. But I think this is the example of strangulated vulva, if that's what that word means. There's no sac. It just leaves these like a ring-like structure at the just above the stipe base. By the way, one thing that was not mentioned by Hunter as one of the key features of section Vaginata is that it's only one of three sections that does not have bulbs. Okay, so in other words, the stipe is totally elongating. So when you cut an Amanita from section Vaginata, if it has a secate vulva, you'll see that the stipe is uniform in its thickness. I mean, it may flare out, slightly at the bottom, like tapering, to the cap, wider at the bottom, but it's not going to be a bulb in any sense. It's going to be uniformly increasing in width across the entire stipe, or is going to be completely equally of equal width across the entire stipe. But bulbs are present in all the other sections other than Vaginatae, Amygdala, and Caesarea. So if you see a bulb, you can already eliminate 3 sections right away.
I'm going to guess that this Pakwan, how do you say pakimpondensis?
Pakimpondensis.
Pakimpondensis has
Pakimpon.
spores because it looks like something that belongs in Starp sorocula
Yeah,
or series ceciliae, and those
Series
as far as I
ceciliae.
know have all
Right.
spores.
So there's another pine barrens. And you see the gen bank sequences for this thing. There's another interesting Amanita section Vaginata that is only found in the pine barrens, and it's called Amanita. What is it? I already forgot the name. Williamsae, named after a female mycologist, perhaps, or some female character whose name was Williams. EAE.
EAE.
Yeah.
EAE?
Yep.
And so only New Jersey pine barrens so far.
It's Williams CAA. See, maybe it's an IAE, something like that. Yes,
Yeah, I
only
know.
in the Jersey pine barrens, and it's very easy to ID. Yeah, that's the one. You can click on that one. Yeah, it's very easy to ID because it has a yellow cap. It's always yellow. It's a very nice shade of yellow. My observation should
Beautiful.
be, well,
Wow, where was that?
that's the one. It's actually tends to be very, very large in size as well.
Oh my gosh, it's so pretty.
So we got the sequence. Unfortunately, the sequence was not complete. It was truncated, and actually, I can, if you scroll down and show the picture at the very end, it shows the
Picture?
chromatogram, which illustrates a point. That's what happens with no, the one before that. No. This one.
Okay.
So this is your Sanger sequencing chromatogram that you don't get with nanopore. You can see if you scroll up, showing the right. The bottom portion of the yeah. You can see that we have a single set of peaks on the left, and then all of a sudden you get double peaks. And this is because one of the copies of ITS inserts an extra character. So one of them has an extra T. So red peaks are T's. So you see there's one extra red where there's a green. So one of the haplotypes or copies of ITS just has an extra T, and because of that, everything is shifted by one character, and then you get this huge mess that you have to resolve, which you can do, and you can get very good at it if you practice. But I can resolve that sequence just by observing that one base frame shift to the right.
Igor, is this a type of indel? Because this is an explanation I didn't understand properly.
Well, that's the thing that you can say that the shorter sequence is going to have a deletion. So the shorter sequence is going to lack one of those extra T's. So we have 5 T's in a row. You can see them on the left, before the black peak to the left. So there are 5 red peaks, and all of a sudden everything becomes double peaked after that. And this is exactly because of that. So an indel basically refers to a deletion or insertion relative to another sequence.
Oh, so it's putting an extra red one in there is what's going on.
So if you didn't have an extra red one, you continue to see only a single set of peaks.
I did not properly understand this.
Right. So what happens when you do the sequencing, everything is already aligned at the left part of the sequence, okay? And once you begin to see a change in length in one of the haplotypes or one of the copies of ITS, then you begin seeing these kind of patterns. And sometimes it shifts by 2 characters, and then you have more of a problem because it's more difficult to separate those two by hand and eye. but you can basically follow if you know exactly what happened. And here I know what happened because it inserts an extra T. You can basically lock into one of these copies and just trace its peaks. Okay, so because everything is going to be lengthened by one, everything will be shifted to the right. Okay, so I don't understand why it shows that black bar across on the bottom. It's really frustrating.
So anyway, Igor, so my understanding still is though that the way that one identifies what would be suspected as an indel or indels is extra characters in the sequence of letters CTAG.
Well, yes.
Or
Again,
extra pair maybe also.
indel is a portmanteau, insertion deletion. Okay, insertion deletion indel. So again, you only talk about that when you have two sequences aligned, and in order to align them perfectly, you have to either insert these blank spaces so everything is shifted, in whatever direction, such that you still have a perfect alignment. So here, if you align these two sequences, and this can be seen on the next slide, you have to click. Yeah, exactly. You see, if you look at the third row from the bottom, there's a G in the lower alignment sequence, but there's a gap indicated by a dash sign there. So this is a deletion. So the upper sequence is shorter because it lacks that extra G. So in order to align everything, and you see here everything perfectly aligned, you have to insert a blank space in the sequence that's shorter. So that's, you can refer to this as a deletion in the top sequence and insertion in the lower one.
See, when you edit a sequence, do you replace that blank with with like an R or a Y or anything
No, you
like
can't.
that?
Dave, I told you, you cannot change anything in the sequence.
But what do you report then? I mean, I
You
don't see
report.
blanks in any sequences on GenBank.
You report a contiguous line of characters, with no gaps. Remember, DNA doesn't have gaps.
Okay, that's I'm sorry, duh. And that's why when you compare two sequences, when one of them has indels or the other has indels, they do not align nicely. Okay,
Exactly.
I see.
Like I said, you only use the indel when you refer to an alignment like that. So you have to compare it with something else. And then I'm going to say the sequences differ because one has, extra characters in it, or that one has fewer characters in it. Okay, so as demonstrated here, an extra G would be an insertion, and the other one doesn't have that, so it has a deletion relative to the other one.
Now, in my experience, this example is a little bit atypical because usually you'll find these indels when there's like, 4 T's as opposed to 5 T's. It seems to
Correct.
be mostly with repeated base pair names.
Yeah, but well, correct. Most of the insertions or deletions, or frequently they come in homopolymeric regions. So when you have 5 T's in a row, and all of a sudden you get another one in one of the ITS copies. But you can have occasionally gaps like that, which almost like seem random. But obviously, since these sequences are complete, and I think they were well edited, there's no question that MO 289538 had an extra G there because it's real. It's going to appear as a
You can
black
see
peak.
that when you see the way the two sequences proceed after the deletion or the insertion, whichever you want to call it, as being pretty much the same after that for a while.
Yes, I mean, they will be the same unless there are going to be any other changes. And this is an empirical result.
Yeah, and of course, when there's too many changes, then it just kind of all falls apart sometimes, I
Right.
guess.
Does it
Again,
ever have?
remember that
Sorry.
this diagram is an alignment of two sequences. You have to show they're the same, but you also have to showcase their differences. Okay, and this can only be achieved by aligning two sequences properly. and a program does it for you. You can do
Can
this
I
in
ask a
GenBank.
crazy question here?
Go ahead, Chris.
How much the same makes them the same? I mean, there's those little tiny differences that I'm looking at. I mean, I don't understand it. It looks like there's a
Yeah,
couple little.
there are single nucleotide polymorphisms, SNPs.
So
That's what you call them.
so like, how many of those do you get before you go, this is maybe something else?
It's not numerical. You have to look at the structure of the DNA at these sequences. So again, these alignment shows that the sequences are not perfectly identical. They do have differences, and this is going to happen. You're going to have different populations of your mushroom from different regions, and there are going to be mutations. It's natural.
Well, Igor, what about this proposal? If you have identity match, okay, between say 98 and 99%, then we can think of that as being perhaps ambiguous. But if you get down into the 97s or below, it would seem to imply, assuming proper editing and aligning, that the species are different.
Well, it's a tricky question. You're going to have variability, okay, intraspecific within the species in terms of its DNA. And what that parameter is, it could be only 0.5% difference. It could be 2.5% difference. There are going to be some populations that are going to have, deletions that are probably, 3 or 4 characters long. It can happen. So, again, this is only a face value comparison. Because a gap is not necessarily counted as a hardcore mutation.
Let's just suppose, for instance, Brandon has a foliota that he put on INAT, and it was sequenced by I think by Steve Russell, and Steve called it Foliota stratosa, and Steve says that he has like 399+% identity matches with other Foliota stratosa. Well, I copy and pasted the sequence into GenBank, and it blasted to a whole bunch of foliotas, but it aligned with the Foliota stratosa isotype, which is as good as it gets. Okay, it was collected by Smith to replace a type specimen that no longer existed. The percentage identity was under 95%. I did a little checking on my own. I copy and pasted the two sequences, the GenBank sequence and Brandon's sequence, into a Word document, and I don't have the software to do the alignment. But I went, a few characters at a time. It took me about 10 or 15 minutes. And it looked like maybe there were a few indels, maybe 2 or 3 instances where indels were kind of, muddying up the water a little bit. But there were quite a few instances where it was just the letters didn't match. The letters didn't match. The sequence didn't change its length or anything like that, like with an indel. And I came up with significantly less than 96% matching characters, disregarding indels. That seems to me like different species.
It could be. My rule of thumb, I don't look so much at the numbers to assign species or say this is, got to be the same as, whatever is showing in GenBank. I look at alignments, or more often, I just begin building trees. So I build a tree in GenBank. It's very easy. You just click off all the accessions that you want to be in that tree, and you click a button, which is, on the website, and it spits out a very quick, distance-based tree. Okay, and I start, seeing patterns, and basically, I'm going to say that you can, if your query sits together in a nice, monophyletic clade with a bunch of sequences that are maybe whatever, up to 3% different, then you probably have a single species most likely. Again, there's no hard and fast rules. You have to have a lot of experience. You have to looking at the alignments and doing trees.
No hard and fast rules, but my understanding is to build a good tree, you very likely need more than just ITS.
Well, I mean, if you are fishing within the same genus, then ITS is good. ITS doesn't work when you're doing a phylogeny of different genera.
Okay,
tree.
now let's throw into the mix a genus like Foliota, where, in my opinion, okay, just as a collector who has looked at a lot of mushrooms, all right, genus Foliota seems to me to be, have a whole lot of like cryptic species. So there might not be database records for a fair amount of what's out there in genus Foliota.
Yeah. I mean, I often look at the sequences that people post in GenBank, in Mushroom Observer, and I plug them into NCBI, blast them. I just want to see for myself, what shows up. And occasionally there are situations when there's nothing than, closer than 95 or 96%. It could be a new species. It could also mean that it just hasn't been collected yet.
So let me provide an example that's kind of on plants, because I'm a botanist first. Let's say you had two plants and one of them has a leaf width of less than 2 inches and the other is more than 2 inches. Is it the same species? Well, it could be two different species and separated by the leaf width, or it could all be one species and it ranges from 1 inch to 3 inches or something like that. Then you add another character like latitude. Some of them, some individuals grow, north of Virginia, others south. Well, if the ones that are all growing north of Virginia consistently have less than 2 inch wide leaves, then those two traits agree with one another. And you add on a third a third trait, maybe one grows in upland or one in wetland. So it's you have differences that are consistent with one another. So when you're looking at different sequences, I would imagine that it's less about the percentage that they differ and more about the consistency of where those differences occur in the sequence. And you can never just look at two samples and say, here's one collection and another collection, and are these different? You want to look at 100 collections of this complex and see if they're separated by a consistent way.
Yes, and that's why you build trees, because trees are going to tell you it's a computational exercise. It's done in silico. This is not something you can do by eye and hand, and it's going to give you a very clear picture. So again, percent identity only tells you how many mismatching positions are out there in the sequence, or comparing sequences, multiple sequences. It doesn't tell you where these positions are. So this is basically Jason's point. You have to look at the alignments and see how different these alignments are and how many consistencies you have between them. Okay, so it takes time and it takes experience to do this, but eventually you're going to start, seeing, for the forest for the trees, so to speak.
Yes, but this also depends on availability of data. With the plants that Jason was referencing, I'm assuming that there's robust data available to compare.
Right. You can go to an herbarium and look at 100 specimens all at once.
Right. With Foliota stratosa, there is an isotype. That, as far as I can see, that's it. That's all there is. And, that's a good starting point. That's a real good starting point, because it's a type specimen. Be it a replacement, it's still a type specimen. And when you compare to something like that, and there's limited data available, and you get 94.3% identity match, to me, that says you're calling this Foliota SP.
Yeah,
I would like to get the discussion back to Amanitas.
Yeah, I was
know
actually
the DNA always will sidetrack us.
I was going to say that as well. I can share the. I was going to share this
Yes.
specimen and hopefully have this conversation later on. But I'm glad
Okay,
we.
well, that's a good point then. Let's if you want to take over the share, that would be great. Is that
So
good for you,
Williamsiae,
Brandon?
is a yellow cap species only found in the pine barrens, and it's usually very large.
And
And it's
does it ever have any veil remnants on the cap, or is it always like
no,
cesaria? Like
it doesn't have any veil remnants on
No.
the cap.
it's amazing looking.
We have maybe 2-3 collections of this thing from FBP.
Wow,
Probably the
that's
same
And-
population, because it's a patch of land, maybe 100 by 100 yards across, something like that.
and massive.
yeah, it's
Big
quite
deal.
large. I was gonna say I could have walked by Lepidella thinking I was thinking it was a lemon yellow Lepidella, but maybe I've been walking past it.
There are no lemon yellow lepidellas. They're all white for the most part.
Oh sorry, I'm thinking of the yellow Amanita.
Floydiae.
What's that?
Something in section Floydiae.
No, the one that turns purple.
Oh, the lavandula.
The Lavandula.
Yeah.
Yeah, but lavandula doesn't have striations because it's a valid.
No, right. But from a distance, I'm just saying I may have just not looked carefully enough.
Those are
That one is-
usually not really big. The ones in the Starps citrina is the
Oh,
rods.
so
Yeah.
some of them can get big though. You'd be surprised.
Oh, okay.
Yeah, I agree.
I'll take Planet Pine Barrens. Okay,
Yeah,
I
I
don't go
agree.
there
I
very
agree
often.
with that. Some of those Lavandula or Lavandula, however you say it, they can get pretty big.
Yeah. So good. I'll keep a lookout for it. That's great. Really pretty one.
So Brandon, do you want to share your picture of your Packin whatever it is, Pakimpondensis?
Pakimpondensis from
Yeah.
Packham
So-
Pond. It's
yeah,
from
yeah.
Without
Packham
it in
Pond.
front of me, I can't pronounce it.
Like Kira was just saying, it's named for Packham Pond in Brandon Burns
State Park. Yeah.
State Park. So if you know Packham Pond, it's just Pakimpondensis.
Yeah, I would have no idea how to say that if I didn't.
But so I have a couple section Vaginatae to share, but also a couple off-topic things. So does anyone have any just Vaginatae things that they want to share before we get into off-topic things?
I was curious about the specimen in the ad for tonight's talk for Taxonomy Tuesday. It looked like it didn't have a species name on it. It was beautiful, kind of orange Amanita section Vaginatae with.
I don't think it was a section Vaginatae, right?
Oh, it wasn't that is that Taxonomy or?
It was supposed to be though, was it not? Yeah, I think it is. I think Igor said that when you clicked on it.
Oh,
Yeah, you're talking about the one that was pictured on the
Yeah.
email that we received.
Oh.
Notifying that Taxonomy Tuesday would be. Yeah, I think that one's one of those weird Vaginatae that has a disintegrating vulva, if I remember correctly. I don't know that one. I have some Vaginatae I could share, but they're from Pennsylvania, and this is such a big, complicated section. There's probably enough stuff from New Jersey to carry the weight of the show tonight. So we don't have to show mine.
Okay, so
Mine
no one knows
is-
what that is, then, so please proceed then, Brandon, thanks.
Real quick, I have one that I think looks like what Dave was showing earlier, and I wanted to confirm. Can I do a quick share?
yeah, definitely. Go for it, Bianca.
Thanks. And also nice to see everyone.
Yeah, good to see you. I didn't know you were here.
I sneak in every once in a while. So this is what I have. Tell me what you think.
Beautiful.
Dave, is this the same as what you were showing?
Rooseveltensis.
That's, yes.
Well, the first thing I would want to do is look at some spores under the scope. If they're ellipsoid, I would say that this is maybe related to rooseveltensis, if not the same species. I'm going to guess not. This looks a little bit more like Fusco zonata to me. I don't know if you get that in New Jersey. But these guys are so hard to tell apart from just with few exceptions. You know, Rhacopus, Texas Aurora, fulva, Crocea, Yamera Crocea, if that really is a name. There's a few others. The deep rooting ones, series penetratrices. Those you can kind of say, okay, I can get in the ballpark with what the species is by the way it looks. But a lot of these grisettes are just very sort of they look the same, and within a given species concept, there's a lot of variability in cap color. So I don't know. Could it be rooseveltensis? I'm gonna guess no. It looks like it's in a forest.
Yeah, that's what I was going to say. And what was the species? I didn't even write it down. Hunter mentioned it as well. You mentioned this might be that.
I think it's called Fusco zonata. It's a new name. It used to be it used to be something else. Igor might remember. There was a Rod Tulas name. It was overridden because Quebec published it as Fusco zonata. Something with E, I think. Not elongatior. That's a different one. I forget it now. Do you remember what Igor, are you there? You remember what I'm talking about, I think. The old name for Fusco zonata.
Yeah, I cannot recall the name. I can look it up and see what the synonym was.
Yeah, I have a lot of specimens that look very, very similar to Bianca's observation from woods.
I think I've seen that too. Bianca, where did you find that?
Was in Turkey Swamp Park.
Fusco zonata used to be what Rod Tulas has a lot of these cryptic synonyms that were there before, but the name they're looking for is longicuneus.
Longicuneus, right? Not elongic. Okay, longicuneus. Thank you. Yep. I had a whole bunch of posts on MO called longicuneus that I ended up having to change when Quebec published the name. And getting back to the three different observations that people are now discussing that they've made in the woods that looked the same, it is definitely not unlikely that you're talking about three different species. It's just really hard to look at these and say what they are. That is why Igor warned the hunter against diving into the deep end of the pool, because eventually you do start to tread water in this sort of environment right now.
I have a good example.
It's a worthwhile thing to explore, really. And plus, there's all these cryptic species that are unnamed, so it's a great like fertile ground for amateur mycologists to go out and try to make good collections and preserve and submit for sequence.
I have a good example that I just posted as a link in the chat of an Amanita that was again growing in the pine barrens that is only native to that kind of a habitat, which is Amanita varicolor, although it's also found in Canada and New England. If somebody could click on that observation, I'm going to show you how variable the morphology is of that species. It's just a single observation where I lined up several fruiting bodies, and they give an impression like three different things or four different things, but they're all the same. This is the same species.
I could say a similar thing about Fusco zonata, the appearance.
Look at the white cap, and if that other, I can, the other photographs you can scroll, across and show the, close-ups of the caps. the vulva can be gray or it can be pink, as you can see on the right.
Right.
Right there. But all these things were found essentially on the same road in the woods, maybe 50 yards, and I think 2 of them were sequenced. So I'm pretty sure it's the same species. And I've sequenced ITS and TEF1, and it came out, to be the same thing. So it's a
Crazy.
highly variable species.
Looks like 6 different species.
Yeah, it looks like, there's an
Right.
amalgamation of,
In one photo, 6 different things.
yeah. So we have 3 pairs of, 3 collections here of 2 fur bodies each, and it's the same thing. Has a strangulated vulva, a chevron like ornamentation on the stipe,
Mm.
and variable vulva, gray to pink or pink to gray.
Like Chiara was saying with the yellow one that we might be confusing down in the Pine Barrens. If I'm looking at the second one from the
Right. Definitely.
That could be crenulata, no doubt,
Yeah.
if you don't excavate it.
Mm-hmm.
Yeah, but again, this species doesn't have a saccade vulva. It just has this strangulated, vulva that appears as a ring, maybe half an inch above the tip of the stipe.
Yeah, crenulata typically has a bulb at the bottom of the stipe. It's a section
right.
Amanita.
Yeah, crenulata is from section Amanita, so it must have a bulb.
Yes, and conversely to what Igor is pointing out here, if they do look the same, they may be different species.
Right.
Yes. That can also be true.
Okay. I think we have other people who want to share some amanitas. Lila, do you want to share yours directly?
Sure, I can do that. Let's see.
Thank you, Bianca, for sharing yours. I don't know if you actually got a name, but got opinions.
Yeah, when you find something like that, try to make a good collection and preserve it, and maybe the opportunity will arise to submit for sequencing, because those are the really interesting things, these section Vaginatae.
Send me a list of pictures of what you want me to find.
Pretty.
Fulva. We got a fulva here.
Yeah, well, very least Starp's fulvae or Starp's. Starp's fulva. Right, it's actually series fulvae. There's a little bit of ornamentation on the stipe here, though. That's a little bit disconcerting. Usually, fulva is pretty smooth. Otherwise, it looks like fulva. And also, usually, fulva has staining on the basal vulva, but that does take. Oh, there's some. Oh, there it is. Look on the left. See the little brown stain?
Yep.
Yeah, it looks like fulva, or if not, something very close.
Okay.
Yeah, it's nice to see the striations on that one because I hadn't seen that before.
That's a
yeah.
good observation. We see relevant details that allow us to have a good conversation about this observation.
Yeah, and even in the young ones, before they open up, that's pretty consistent, right? The striations, even when they're pretty immature.
Usually with Vaginatae, you can see the striations early on. In section Amanita, oftentimes the striations do not appear until the cap is expanded, like in Velatipes and the muscaroid ones. And sometimes in the muscaroid ones, you hardly ever see any striations, even when it's expanded. But Vaginatae and ceciliae, the striations are usually fairly observable from the start. Mike's, that's Mike.
Okay. Next
Yeah.
one.
We could ask, why doesn't iNaturalist figure it out to the species on that one?
Because AI sucks.
What
There's no substitute for the human eye.
Yeah, but
yeah,
what
but,
do you
but,
call
but
it?
iNaturalist, if they called this section Vaginatae, then they're just being honest.
Yeah.
We don't know what this is based on.
Honest or incompetent.
Well, well, what would you call? Do you? I
This
mean, is
thing
that?
here.
No,
The-
no, I was
the-
talking about fulva, not this thing.
Do you call it vulva? Is that vulva proper in your opinion?
Oh,
Yes.
that
Yes.
last one. Is it? Does it have to be fulva?
It has to be fulva.
Okay.
I would wonder. a
Rather,
little
you
bit
figured
about
it out.
something.
It's all fulva.
Well, that's what Steve has been saying too. So whatever happened to this species 02 or whatever it was? I guess that got absorbed into fulva.
Got absorbed. You know, there's
What
a
about
North
that one
American.
that started with D and has like too many syllables? GAIM, blah blah blah. I forget. I guess that one got absorbed into fulva also.
Yeah, there's a Eurasian fulva and North American fulva, but you know, as far as the DNA is concerned, you know, probably ITS for the time being, it is the same thing. Okay, so this thing you need to sequence.
Yeah, that's what I was saying. Yeah,
Uh,
they're just being honest when they say section vaginatae, and you can't say much more.
So you do think it is in Vaginatae?
it is definitely section Vaginatae.
Okay.
By the way, Amanita Vaginata, the European one, there is no type collection. Nobody knows what Vaginata is. So, you know, when people tell you in Europe that we collected Vaginata, they're full of it.
Well,
There's no
what
type
sort of
specimen.
what sort of data exists for the gray grisettes in Europe? I mean, is there a lot of variability in the DNA, or is it pretty homogeneous?
I
mean,
don't know. My question is, you
that's
know,
what I'm saying. Yeah, I don't know either. But if it's
if you have
pretty
several species,
homogeneous, if it's
if you have
pretty
several
homogeneous,
species,
what's that?
if you have several species, you have to assign a Vaginata. You know, one of them has to be Vaginata, right?
Yeah, but if the data is very homogeneous and they're virtually all matching DNA wise, then you can probably with fairly reasonably good confidence in Europe say, oh, these gray ones are vaginata. But if the data is spread around, you know, and suggesting several different species, then what you're saying, I would agree with. Then
I'm pretty
nobody in
certain.
Europe knows what they're talking about when they call it vaginata.
I'm pretty
In that,
certain that there are several gray-capped species of section Vaginata in Europe.
okay, that's sort of what I'm asking because I don't know.
So now this Amanita, where
Was
was
this found
this
in
found again?
moss?
Probably in my yard.
My guess, this Amanita could be species QEE4, Quebec 4. And I'm saying that because it's very long, has a very long stipe. It has a powdery internal limbus internus from the vulva at the upper stipe, just like rooseveltensis has that. And the cap is gray. So I have an observation of species QUE4 or O4 from one of our forays. What's that one? The horseshoe bend.
So this was found in a yard, though, an open area.
Well, it's my yard, which is a lot of large oaks and hickory and white pine.
But is the area fairly open? Gets a lot of sun.
Well, I don't know. This was 2023. I can't remember exactly where in the
Okay,
yard
because
I
the,
found it,
because
but
an
you see all this moss, so it's
right,
not a lot of sun.
because an open area would suggest rooseveltensis, and sometimes rooseveltensis, as Igor just pointed out, has the floco stipe. I mean, I found plenty of them that look like that.
Yes, one of the features. I think all the rooseveltensis that I found in my property, and I'm pretty sure they are rooseveltensis based on the appearance and the spores, which are ellipsoid, have these sugar powder at the upper half of the stipe.
And
Does that
have you ever?
Q thing have ellipsoid spores? Do you know, Igor?
QE4, what has ellipsoid spores, I don't know, but
Uh,
like I said, we found a collection at the Horseshoe Bend a couple of years ago, and it looked interesting, kind of reminded me of rooseveltensis, and when I got it sequenced, it turned out to be that QE4.
Interesting. So it would be interesting to know the spore morphology of these then, so that you could know without sequencing, have a pretty good idea, yes or no, regarding rooseveltensis.
I'm glad we talked about rooseveltensis earlier because I never even look at the spores. I do analytically test if I have to, if I don't know what it is. But I never really even look at the spores because they all,
They all
all
look the same. They're all globose. But as soon
Yeah.
as you run across one where they're ellipsoid, then you
didn't
know
even
you
know
have
about
something
that. So
interesting.
that's good.
Yeah, speaking of spores, one thing I forgot to mention about varicolor, that the spore is also going to vary from almost globose to broadly ellipsoid or ellipsoid. So not only that species is variable in its morphology, it's also variable microscopically.
This looks like elongatior to me, at least the ones that I've picked. Is the stipe pretty deep? In the ground, it looks like it is. Was this in oak? Oh, okay. So you had to dig it up a little bit. Were there oaks in this area where this was
I'll have to
found?
look and see where I found this one. Hang on. Let's look at it first.
Thick vulva. That's
Yeah,
another
look at how
interesting.
like almost looks like a piece of rubber split open, right?
Yeah, some section Vaginata have very thin vulvas. Other have much robust, hard shell, thicker vulvas, and I think it's important to notice these kind of distinctions.
Yeah, I would have to review, but I think my observations of elongatior, which were sequenced by Rod and Linus, have a rather thick, persistent vulva, and it tends to be buried at first. So when they are buried at first, the shape can affect the shape because it's pushing up through soil and debris and so forth.
I think that
Lila, that's a really good observation.
Thanks, Brandon. I'll have to log in to see exactly where it was that it was found, but it was back in the good year of 2023.
By the way, elongatior is a published species. It's available on INAT if you want to propose it.
So why was this
in
identified
yard.
as vulva?
Okay, here it is. Here it is. Where? All right. So this was in my backyard. Mature pin oak at the edge of a wood chip pile.
Well,
But the ID says it's fulva. This is not fulva.
no, that's not fulva.
Not fulva. Okay.
No,
I
it's
don't
not
know what
fulva.
it is. What do you think it is? Elongata?
Elongatior. I'll put it in the chat.
Okay.
Oh, it's just a guess though. When you're going on morphology, you're guessing. But it looks. Here's what looks like elongatior to me. At least the elongatior I have found, and I believe that Igor has found an albino one of these.
Yes,
But
it was sequenced.
do you see how the disc is dark? And do you
Yeah.
see how there's what they call innate fibrils radiating toward the away from the disc? I had told Rodtulas before this thing was named that it reminded me of what it might look like if you saw a comet coming straight at you with like the debris streaming off the comet. And he really liked that name and he called it Cometa Adventia or something. I forget. It was a lot of syllables.
Cometa.
Adventia Cometa, right? They had approaching comet, and he loved that name. It was like it was more of a bummer for Rod than me when Quebec published it. Didn't use his name. So that's what I'm seeing here, especially in the first photo. You can see these innate fibrils. And innate fibrils they're not on the cap surface. They're just like embedded inside the cuticle. So it looks like fibrils, but you can't actually rub them off or anything. They are innate. Do you see how it's like radiating from the disc?
Yeah, definitely.
Yeah. So that reminds me of my observations of elongatior, which you can find on Mushroom Observer. Oh, you know what? I put a few of them on INAT recently because our Pennsylvania person who's in charge of the newly designated position of conservation of fungi, she wants to make Hannah Huber. She wants to make a list of all species found in Pennsylvania, and I have a few of these that were sequenced, so I'm confident about them. So she uses INAT. She bases everything on INAT. So I put them on INAT recently. So you will find that name on INAT.
Okay.
And I'll put it in the chat.
All right. Is there an easy way to cross share from INAT to Mushroom Observer, or do I need to like reproduce the whole thing? Does anybody
No.
know?
You can import observations from INAT into MO. There's a gizmo.
There is.
Yeah, there is. Yeah.
It works really well. I just used it because I was actually having trouble using MO yesterday. Like they changed some sort of interface or whatever you call it. All of a sudden it didn't want to recognize the dates of my photos, probably because the photos are taken with a camera and not a phone. And so actually I ended up making a few observations on INAT and importing them to MO. And until I got a little help from tech support on MO, and found out what I should be doing, which is a little bit of extra work. It's not much, but it's easy. It's easy to import from INAT to MO.
Right.
You started on MO, right? And use that import.
No, you started on INAT and you go to and you copy the INAT observation number onto your clipboard. And there's a thing on MO now that says import from INAT, and you click on that, and it asks you, it asks for your permission to do this, and the usual nonsense.
So okay, cool.
And then you just paste in the INAT observation number, and imports the whole thing for you, including the name that's proposed.
That brief explanation troubleshot everything that I've been trying to do. So
yeah. Well, I think this is recent. I think this is fairly recent with them, but it's easy. I was able to do it. It's like Mikey eating the cereal or something, right? When Neanderthal me can figure out something like this. So
see, that's where I got it. Neanderthal.
So when you say the INAT number, you mean like the 8-digit number, not the whole URL, just the 8-digit number?
Still only 8 digits?
Well, 9, whatever it is now.
Yeah, right.
Yeah.
You know what I mean. Yeah, that thing. Not the whole web address. You just paste in the number.
Okay, cool.
And it takes like 10 seconds and it's there. Now, you will get the name that is on INAT and it's going to say, I'd call it that, which you might feel rather dubious about. You can change it very easily. You just propose a new name on INAT, vote on the name they give you. You can downgrade it to the various levels of confidence available. And all of that is pretty easy.
All right. When I have some time, I'll try to do that.
Of all the Vaginata species I looked at this last week, the elongatior had the most substantial vulva of any others I looked at, except for the West Coast choice edible, maybe. But
that's what I thought I remembered, Hunter. And it's on top of that, the vulva is usually at least partially buried, so it's got to be pretty persistent and thick, to stand up to being buried and then getting excavated.
Yeah, really substantial.
Okay, thank you, Lila. Does anybody else have any Amanitas to share besides Dave? I know he's got a list. I don't know if we got everything from Brandon.
If there are more New Jersey observations, I defer to those because that's certainly more relevant to most of the people here.
I have one from this week.
Okay.
Whoa.
Do you know how to share it?
Yeah, I learned it from you guys from the newsletter.
Go ahead and do it then, if you can.
I'm looking for, I don't use, let's see. Let me go to I don't use the desktop version of iNaturalist very much. If you guys tell me how to find my pictures. Here we go. So I read on the newsletter about the diatrite fluorescens.
You got to start sharing, Kiara.
Yeah.
Oh, I thought I did that. Sorry.
Oh, cool. Another diatribe fluorescence.
Yeah,
Did
did
you
you?
report that to MO? Because I saw one recently from New
I did.
Jersey.
So
Yeah,
it
I just
turns
voted
out,
on that. I said that's what it is because it certainly looks like it.
can you see my screen yet?
Not yet.
Not yet.
I'm clicking share. Oh, wait, hold on. Okay, sorry about that.
Did you pick the stick up off the ground?
Yeah, and you know what? I run a camp. We have a 50-acre forest, and most of it seems like it's beech forest.
I
And
was going
it's
to
under
ask if you found those all in one day that you
it's under
uploaded.
every tree, Brandon. I've actually found it under probably 20 trees so far. It's under every single tree.
Yeah, makes
So
sense.
apparently it's also an endophyte. It lives within the branch when it's alive, and then it turns into a saprophyte when it falls falls to the ground.
Those are nice.
I'm guessing that it actually, as an endophyte, actually lives inside the leaves
Oh, you think so?
because that's what some of them do. Yeah, that's what some of
Interesting.
that's it. This is all pretty recent in my
Mhm.
experience.
Okay.
Is that some of these endophytes are living inside leaves and then they become saprophytes on some of the more substantial material available in the organism. So in this case, a stick. Were these mature beech trees in this forest
All,
where you found them?
different ages. Some very young beech trees and some mature beech trees, but I'll try to make a closer observation of that next time. I was actually going to measure the tree.
That's hard to do because if they're on the ground, who knows what tree they're falling from? The wind blows things around.
True. There, I have one area where it's all beech forest, so it is hard to tell. But in another area where I have a few separated by oaks, so I can definitely tell.
Good. So you have like a control group.
Yeah.
Sort of.
But anyway, under every single beech tree I checked, they were there. So
Because I
funny.
just checked it here where I live, where there's beech, quite a few beech, but they're pretty young. And apparently these young beech were like the offspring probably of this really, really, really old beech that was cut down probably about 10 years ago, and the logs are lying around. And I couldn't find this fungus anywhere in that area. They were all young beech trees.
All right, so how do I stop sharing? Here we go. I see it. Sorry. Just that one. I figured you guys had seen it, but I learned it from the newsletter from last week's email. So thanks.
Nice. They're fun to find. They're so tiny though.
They are. And
so
I've seen them so many times. I just, overlook them. But I've
Is
taken
that
pictures.
INAT observation research grade yet?
What's that?
Yes.
Is it research grade?
Oh, I'm not sure.
Yeah, it should be. It
Okay.
should be.
Yeah, I think they.
Oh,
Whenever I check, I checked my local fungal INAT finds, and I saw she uploaded like 4 or 5, and I think they were all research grade. You found them all in one day. It's so good.
Oh yeah, I found more than that. I just haven't uploaded them yet.
All right. Do you guys mind if I start sharing
Oh, okay.
pakimpondensis?
Just a second. Yeah. Do that. Go ahead, Brandon.
All right. So let's see. I think it's this one. Yeah, so this is the one that got sequenced. And I've shared it here a bunch of times. And we've all pretty much speculated, yeah, it might be pakimpondensis. But, sequence came back. And one of the things that tells me immediately, oh, this might be a Rhacopus or pakimpondensis type thing is that gray UV. Very much gray in color on the cap with the striation that is in line with everything that Hunter presented earlier. And you can see that UV material is pretty gray.
Yeah, now back to the UV material. Well, I took the tidbit about there might be a patch at the top, but clearly some of these have a lot of velar material all over it.
For pakimpondensis species specific or non-prov, this is a non-prov or group or whatever it
This
is.
section. This is still Vaginatae, right? Is pakimpondensis in that section? Yes.
Yeah, only some of them have that centralized
Okay.
UV. Correct me if I'm wrong. Again, this is not my area of expertise, so Dave or Igor can maybe correct me there. But some of them, that UV is going to be more centralized, but a lot of them have patches, and this is kind of more marginate.
Yeah, well, even rooseveltensis that I'm pretty familiar with will occasionally leave some patches on the cap. Usually it doesn't, but it's not, definitely one way or the other.
Okay, so it can have patches in other places other than the one big one or whatever. Okay.
And sometimes no patches.
Yeah.
Mhm.
Yeah, more often no, probably, but this one tends to have it, I guess. Pakimpondensis. Cool.
Are those gill edges a thing?
Oh no, Chris. Yeah, I was just about to ask that because I haven't done a lot of microscopy on these things because it always kind of is. Well, there's not a lot of information about the microscopy, so what's the point? But now that I'm starting to pay attention to them a little more, like, yeah, we need to start paying attention to these things. And you would have to assume that the gill edge is being tattered here, or maybe that's chylocystia.
No cystidia on Amanitas.
Okay, universally no cystidia.
I'm pretty sure that's true.
Okay, but yes, these gill edges are.
There are other structures found on gill edge. I think there might be some things they call terminal cells. They look like balloons, kind of like, but they're the tip. They're for some technical reason they're not called cystidia.
Okay.
But Igor mentioned before the limbus internus sometimes causes deposits on the stipe, and I think some of those deposits can be transferred to the gill edges. I'm not sure about that. Igor, you have a thought on that?
Yeah, well, the reason there are no cystidia on the gill edges is because the gill edges are fused with all the other tissues in the primordial Amanita, and when everything separates, everything kind of rips apart. There's some tissue that allows for the cells to break, between different, parts of the internal parts of the Amanita. That's why the gill edges of Amanita are sterile. There's no pleurocystidia or chylocystia in them.
Okay.
It's just the way Amanita develops. Its ontogeny, its development is very different from all the other gill mushrooms.
And you can kind of see that with the striations at the apex of the stipe here, right? That's pretty much what we're seeing, where the gills broke away abruptly from the stipe apex.
I'm not sure about that particular feature. I think other mushrooms have that same, those ridges too. But these gill edges were it's kind of difficult to describe. You have to see when you cut an Amanita egg, you see a silhouette of an Amanita, but it's like completely solid tissue all within. But again, all the elements of the gills, they are there's tissue on them that allows the structures to break away from each other without tearing itself apart. Okay, and again, the reason the gill edges are sterile is because they already fused with all the other parts of the mushroom inside that primordial or primordium. And when things, when Amanita grows and expands, all these tissues come separated and they have to break apart from each other cleanly. Otherwise, they would just tear itself apart. All right, and this is what you're seeing here. You see all this powder. This is the remnant of all these all that special tissue, specialized tissue that keeps all these elements separate in a primordium, but allows them to form an intact mushroom at the same time.
All right, cool.
That's a really good explanation, Igor. Thank you.
Yeah.
Yeah, again,
understand all
it's
those
tough.
details.
You have to read what Rod writes about it on his website, and he's much more eloquent about it. But I haven't seen it in a long time, so I'm just trying to paraphrase what I presented 10 years ago. In that presentation that I've been talking about. So
Right.
who sequenced this Amanita for you? Rockefeller?
No, this was Fundus.
Oh, Gibbs found this. Okay, got it. Good.
Yeah.
Cool.
Yep, that's Byron.
Yeah, I turned off the mute, just for you, Igor.
Cool, cool. And we'll
Byron,
just
your
look at
buddy,
real
Uncle
quick,
Igor.
because I don't know if this is actually Rhacopus or not. But it has, it's more, it's a little bigger than a lot of the section Vaginatae that I'm used to seeing.
Vulva's
Tall
not
and
turning
slender.
gray. The vulva's not turning gray on the bottom. So I think this is something else.
You sure?
Oh, I'm sorry. Up, another picture. Okay, yeah. Nope. I retract that previous statement. Stirpsirocula, I suppose.
Yeah, I think that's what I put on MO. I just it's easier for me to upload photos here, and you can see, Dave. I'm glad you said no originally because it's only the underside of this that was gray.
Yeah, it's not notably gray either. Usually by the time it's this well disintegrated, that upper part of the vulva that persists is pretty gray. But look at the patch just above the ring. That's kind of gray. I mean, maybe this one is actually pretty young and the vulva just fell apart rather quickly and it's not yet really strongly gray.
I wouldn't be surprised if many or maybe all of the Amanitas in Stirp ceciliae have this friable strangulated vulva because it,
Yeah, I think that's the distinguishing feature. It's series ceciliae, I believe.
yeah,
I
series
think that's the
ceciliae.
distinguishing feature. They are the strangulated amanitas, I think.
So that's
Yeah,
what you were explaining earlier, Dave, right there.
Yeah, it's a good example. Sometimes it's not quite so. I mean, this thing is almost, like a rim, plastered along the entire perimeter of the stipe surface exterior. Sometimes there's just like patches and it's a little bit disorganized, but there will be this ring of UV material on the upper part of the base of the stipe that strangulates the stipe on the bottom.
Can you guys say the series that you thought this was in?
Ceciliae.
Ceciliae.
Yeah, just like a feminine name, Ceciliae. So this is going to be Ceciliae, off Cecilia. Cecil.
Yeah. Cecil.
With C's.
I just put it in there. I think I spelled it right.
All right, cool.
I put it in the chat.
All right.
Now, can someone tell me what a series is versus a section? Okay, maybe not.
It's just a
No,
more
slurps.
narrowed.
Slurps first.
It's a more narrow body of, it's a taxonomic unit. Okay, so it's smaller than section, but larger than
Okay.
stirps.
I don't
Okay, so this
think
is still
that these
Vaginata
distinctions are
section, but then it
necessarily-
goes this, okay.
I don't think these distinctions are necessarily supported by DNA data. I
No,
think
not
they're mainly
necessarily.
based on morphology. Yeah.
Yeah, this
Right.
is still section of Vaginatae, as we can
Okay.
tell by
So it's not a
striated,
subsection,
cap.
it's just something separate, another made-up name.
Right,
Or
no
is it-
no ring and no basal bulb.
Anyone care to share another Amanita? I'm gonna break code and go to something different.
Yeah, I'm sorry. I thought we had done that when he said no more. He wanted to focus on New Jersey. I jumped in with the non-Amanita species. Sorry about that.
Yeah, no problem. We want to look at
I know
everything.
Dave's
and
got plenty
anything.
of these
No, that's all
Amanitas.
right. We should look at this New Jersey stuff.
Okay.
Couple from.
Well, I really
Denver
want to.
Pines.
I really want to ask about this because I've
Oh yeah, you
never
showed me
found-
this. This is great.
Yeah, I've never found this thing before. These are tiny, like tiny, tiny. I think Dave might be my only hope into. Have you ever found something called Lachnellula? Like
I don't
Lachnellula?
think so. I'm gonna, you know what? I'm gonna go away for 5 minutes, check my MO observations, and do an advanced search and see if anything comes up. But I don't think I've ever found these things.
What was the substrate that you found
I'll
these?
see.
Yeah, so this was on pine, specifically pitch pine, and the species that I actually think it is Lachnellula resinae, maybe it is, and that's supposed to grow on pine sap or pine
Wow.
resins.
Cool.
Is
So
that like some old little deflated ones there?
well, that's a good question.
I don't know what else word to use. They kind of look deflated.
It's like
I'm
a
not
little cup fungus.
Yeah,
Yeah.
you're talking
But
this here,
almost
right?
not.
Well, they look very crystallized, which is a very, resinous thing, right? Doesn't some of this look like kind of irregular sap, maybe?
Was this on a stick or on the trunk? Like what part of the
So
tree
this was on a fallen branch.
branch? Okay.
But, look at that beautiful yellow cup. I mean, it's an egg in a hole. Isn't it?
Spelling on this again? Sorry.
Like Lachnellula.
Lachnu. Okay, Lachnu. Okay, that should be enough to get me.
And is the background on that is that actually sap?
Lachne.
Oh, sorry.
Yeah.
The shiny stuff on the background of the picture is that sap?
I couldn't make it out. I mean, here's why I couldn't make it out. This is the size of them. This is 1 millimeter.
Okay, interesting.
So these were between maybe a third, and this piece of bark was raised up a little bit. It was kind of concave. So between a third and maybe a millimeter.
It looks the cups look they're striate and they're a little powdery looking.
Correct. Yes. So this is an in situ photo.
Wow, they're great.
Pine branch. Now, I was interpreting this black as just kind of moisture because that's where it was set, kind of somewhat buried. But it follows up here, and I still think that's just kind of moisture, but I don't know how sap actually works in the wild. Gosh. But yeah, like you were saying, kind of a tightly packed, not quite hairs, more just
Yeah.
fluffy,
Yeah, it's very
if that makes sense.
powdery. Hmm.
Yeah,
Kind of got that
powdery.
look of like the origami hasn't gotten all the way there yet.
Well, yeah, and the other part about it is kind of found it in xeric conditions. You know, it hadn't. There was some moisture there recently. I tried to rehydrate these, and they did open up a bit. Like this one right here opened up to the point of that.
Yeah, they are cute, like little eggs. Definitely.
So you know, they open up, they close. The microscopy was a total bust. I got some asci, um, so I know they're ascomycetes. I mean, obviously they're little discomycete type deals, but they were either all immature, so lacking spores within. I didn't find a single spore, or they had already expelled them the first time that they open up, and then they went dormant. I
Well,
don't
they're
know.
so little, they probably only had one spore each. Just kidding.
Yeah.
So this is,
I
I found
wonder.
an observation on MO of mine, and it was actually it was proposed by Jason. I put the link into the chat, so it's in Mushroom Observer observation. If you're not a registered user of Mushroom Observer, you won't be able to see it. But Jason, is this a lichen or a lichenized fungus or something like that?
Oh, wow. I feel like I've seen that's like a disc fungus. Is it a cup? Interesting.
Well, every time I find one of these little things, and surprisingly, this wasn't growing. Well, actually, there were several tiny thallus immature lichen on that stick, but I didn't think they were associated. But every time I've been finding these little discomycetes, little ascomycetes, that relationship between you know, what's going on here? Is it lichenized? Is it parasitic on the wood? Is it parasitic on another fungus?
Yeah.
A lot of the times in the paper that I'm reading, it says that something between symbiotic and parasitic cannot be ruled out, right? So, Jason, are you here? Is this a lichen, Lachnellula? Lachnellula?
Jason's dropped off. It's 9 o'clock already.
Ah.
Interesting.
Yeah, that was cool. And then,
Can you share one of your pictures in the chat?
one of mine?
Yeah.
Yeah, sure. This other thing, I have no idea what it is. You want a picture of this thing?
Yeah, that would be great.
I'll just throw the.
I guess I can find that on iNaturalist, actually. Don't worry about it.
No, I'll just throw the observation in here.
Thank you.
No problem. And then I'll be quick on this because it's probably only cool to me.
Well, it's
Here's
not as
another-
easy to find as kiwi bubbles.
Yeah. Oh, yeah.
Well-
You found this on Friday.
Yeah. You probably have a better photograph of it than I do.
I think this is pretty good.
But they're tiny. I mean, when I say tiny, this is what they looked like on the little tiny. I'm calling it a pine twig. I think it was on pine. Kiara,
I think that's oak.
you think it was oak?
Doesn't that look like an oak bud
It
at the
does.
top?
The bud at the top
Yeah.
definitely does. Yeah, you're
Yeah, I
right.
think it's oak. I see 2 more buds there too.
Okay, so this is something found at Franklin Parker on Friday.
Correct. Yeah. Yep. Found this at Franklin Parker. Probably on oak. I put pine in the description of this thing, but I think you're right on that.
Yeah, I remember thinking that when I saw it too. I didn't mention it.
but I really thought it was one of those ceciliae fungi that Jason gave us that really great presentation on. But even in that last photo, you could see that it was more kind of disconnected at the top, right? It wasn't like a perfect disc or a perfect pinhead or anything like that. Kind of separated at the apex.
Yeah. Mm-hmm. Little hairy guys.
Little hairy guys. Yeah. Like little wild head.
Funny.
This is at 100 magnification. So yeah, it's uniform without. I didn't find a single ascus or anything like that. The only spores that I found appear to be conidiospores growing off of these conidia that are, wildly irregular. They are septate and they're simple septate, so they don't have clamp connections. The things that look like clamp connections, like right here, that's just an irregularity in this otherwise straight thing. Here's a conidiospore being birthed.
That's great. Oh my God.
Yeah, right off the top, right off the tip of the conidium.
Oh, I love that.
But yeah, I truly have no idea what this thing is. Here are the conidiospores. They're kind of avocado shaped. They are thick walled. But yeah, that's all I got on it. I'm just calling it something in Ceridiomycetes or Ceridiales, really. I have no idea what the thing is.
Very cool. Really nice pictures.
Thank you. Anyone have an idea on what that thing could be?
Awesome.
Too
Yeah.
boring, and it's just plain ordinary gray, right? Gray or black. Does it have any coloration at all?
It's black, Sue. Yeah, cool. Thanks for letting me share, guys. Good meeting too. That was a fun one.
Great.
Sorry we didn't get to all the Amanita species that Dave has. Those who have more time can go search for Dave W. on Mushroom Observer.
Yeah, if you go to Mushroom Observer, and you do an advanced search, the advanced search feature works really well. You get several fields to fill in. You do not need to fill all of them in. Into the name of the mushroom field, fill in capital A, Amanita, lowercase s, S-E-C-T period, capital V, Vaginatae, V-A-G-I-N-A-T-A-E. And in the observer, there's a field for the observer. Just capital D-A-V-E space W. So I got really tired of having a long name that nobody could ever spell the right way. So on Mushroom Observer, I just called myself Dave W. So good choice, because it makes it easy to do this kind of stuff. And you'll find like lots and lots and lots of observations. And I think it gives all of them that were at one point named section Vaginatae. So that's quite a few, because most of them started out at that level of generality and then perhaps moved on to more specific names. I think that's how that search will work.
Okay, thank you, Dave. And thank you everyone who shared, and especially for Hunter for creating the startup presentation for us.
Yeah, that was
You know,
great.
happy
Thank you.
topic.
Yeah. Big up, Hunter. Thank you.
Thank you guys.
Okay,
Thanks for bearing with me through it. It was pretty painful for me, I gotta say, but
Oh, it was so good. It helped me a lot.
all good.
Hunter, that was really good, because everybody, including me, learned some things.
Yeah.
And
Yeah.
when you delve into an area like this where it's not all cut and dry, that is what is probably going to happen. And
Yeah.
that's a good thing.
Good, good. Yeah.
And I hope, Hunter, that no one learned more than you. You know,
I don't
because
know.
every time I've ever done something like that, it's like, oh, I didn't know how much I didn't know, and you
Yeah.
Oh,
present
yeah, I
that.
feel like that every day.
Yeah, no,
Yeah,
I
exactly. We all do. You present that to the rest of us. You did a great job. I appreciate it.
It's great.
I enjoyed it. It was a little intimidating, but I did throw a observation of a ringed section vaginate from Canada,
It's in
cingulatoides.
the chat.
And it's in the chat. It's like, because there are one or two that have rings, and I forgot to say, so, but that's that. So looking forward to next week with everyone.
All right.
Right, thank you.
Thank you, everyone. Have a
See you
good night.
later.
Bye.
Good night. So Brandon, if you're going to hang around, if Igor hangs around also, we could probably
Yeah.
go into more depth in this Foliota thing and maybe sequencing in general. And anybody else